Hello guys, in this session we'll be dealing about synapses present in the central nervous system. It is a very important topic and this topic forms the basic knowledge for understanding the central nervous system. We'll be seeing the introduction, types of synapse, anatomy of synapse, mechanism of synaptic transmission, synaptic potentials, electrical synapse, properties of synapse and neurotransmitters. First, what are synapses? Synapses are neuronal junctions through which information from one neuron passes to the another neuron. The neurons taking part in the synapses, the neuron which sends impulses are called presynaptic neuron, and the neuron which receives impulses are called postsynaptic neuron. And through synapses, neuronal messages are conveyed to appropriate structures in the central nervous system. Next, looking into the types of synapse, it is broadly classified into anatomical types and physiological types. Anatomical types is based on the structures involved in the synapse. First, it is axodendritic synapse. Here, the axon of presynaptic neuron forms synapse with the dendrite of postsynaptic neuron, and it is the most common type. Next is axosomatic. Here, axon of presynaptic neuron synapses with cell body of postsynaptic neuron. The third type is axonic synapse. Here, presynaptic neuron, axon of presynaptic neuron communicates with the axon of postsynaptic neuron. The last type and the least common type is dendrodendritic synapse. Here, dendrite of presynaptic neuron synapse with the dendrite of postsynaptic neuron. Moving on to the physiological types, it has three types. The chemical synapse, here the impulse transmission is through release of neurotransmitters. And the electrical synapse, the adjacent cells are connected through gap junctions which will allow the movement of ions, and impulse transmission is taking place through the movement of ions. The last type is conjoined type, which is partly chemical and partly electrical. Next, moving on to the structure of a chemical synapse. As I said, a synapse involves a presynaptic neuron, synaptic cleft and postsynaptic neuron. The terminal of presynaptic neuron ends in a small bulbous enlargement and is devoid of myelin sheath, and this bulbous enlargement is called synaptic knob or terminal button. It contains two very important structures. Synaptic vesicles are rounded structures or spherical structures which contain neurotransmitters in it, and mitochondria which provide ATP for the transmission of neurotransmitters from presynaptic to postsynaptic neuron. And see, this is the presynaptic or synaptic knob which contains mitochondria and numerous synaptic vesicles. Next is the presynaptic membrane. This part is the presynaptic membrane. It is the membrane of presynaptic neuron. It contains large number of voltage gated calcium channels, which is very important for exocytosis of neurotransmitters, that is calcium mediated exocytosis. Whenever it is opened, it allows calcium influx and there is exocytosis of neurotransmitters. Next is the synaptic cleft. It is the gap between presynaptic neuron and postsynaptic neuron, and it is filled with extracellular matrix. Next part is the postsynaptic process. It is the part which synapses with the synaptic knob. The last part is the postsynaptic membrane. It is the membrane lining the postsynaptic process, and this part is called postsynaptic density because it contains large number of receptors for the neurotransmitters released from the presynaptic neuron to act upon. Let us look upon the neurotransmitter receptors. There are generally two types. One is ionotrophic receptors. It means that they directly gate ion channels and allow passage of specific types of ions. While the second class is metabotrophic receptors. These receptors act through second messenger system, that is mainly G-protein coupled receptor. Okay, let us see in detail about each of them. First is ion channel receptors. There are two ion channels. One is cation channels and another is anion channels. What is the function of cation channels? Cation channel conducts, whenever cation channel is open, it conducts sodium ions into the cell. So, a neurotransmitter that opens cation channels are called excitatory neurotransmitters. Let us see reason for this. Whenever it opens cation channels, sodium ion enters and the resting membrane potential of the nerve will become less negative. That is, if initially it was minus 70, it will become somewhat like minus 45 or so. That is, it is going to depolarization and there is excitation of the postsynaptic neuron. So a neurotransmitter that opens cation channels are called excitatory neurotransmitters. So in case of neurotransmitter that opens anion channels, for example, chloride moves into the neuron and the resting membrane potential becomes more negative and it is towards hyperpolarization. That such neurotransmitters are called inhibitory neurotransmitters. Second messenger system, it is mainly through G protein coupled receptors. The receptors are coupled with G protein, which consists of three subunits, alpha, beta, and gamma. When it is in inactive state, the three subunits are together and it binds to GDP. Whenever a neurotransmitter substance is binding to the receptor protein, there is a conformational change such that GDP is transformed to GTP and alpha subunit dissociates from beta and gamma subunits and causes the specific effects like opening of channels, activates one or more intracellular enzymes, activates gene transcription, and activates certain enzymes like cyclic AMP and cyclic GMP. Moving to the mechanism of synaptic transmission, the mechanism is divided into two parts, presynaptic mechanism and postsynaptic mechanism. And let us look at presynaptic mechanism. Whenever an presynaptic neuron is stimulated, or whenever an action potential arrives at the presynaptic neuron, sodium channels are opened and whenever sodium channels are opened, there is depolarization. This depolarization of the terminal membrane causes opening of voltage-gated calcium channels in the presynaptic membrane. As a result, there is calcium influx and intracellular calcium increases and there is calcium-mediated exocytosis of the neurotransmitter vesicles. The neurotransmitter substance is released into the synaptic cleft. Now, after release into the synaptic cleft, these neurotransmitters diffuse and bind to the neurotransmitter receptors present in the postsynaptic membrane and causes the opening of certain ion channels and whenever it is causing depolarization and it causes action potential is fired. This is the mechanism. Let us look into its summary. First, action potential arrives in presynaptic neuron. Depolarization causes opening of voltage-gated calcium channels, calcium entry, release of neurotransmitter via calcium-mediated exocytosis, then binding of transmitter with the receptors, then opening of ion channels, movement of ions through postsynaptic membrane, and change of membrane potential in postsynaptic membrane, and there is generation of synaptic potential. Okay. This picture will explain you. See, whenever depolarization is there, there is opening of voltage-gated calcium channels, releasing of neurotransmitter, and this neurotransmitter will bind to the receptor in postsynaptic membrane. That's all. It is very simple. Okay, what is the fate of neurotransmitter that is present in the synaptic cleft? Either it has to undergo degradation by the enzymes present there, or it will be reuptaken by the presynaptic neuron. It has two fates, either degradation or reuptake in the presynaptic neuron. Okay, let us look at synaptic potentials that are generated in the postsynaptic neuron. Generally, there are two synaptic potentials that are generated. One is excitatory postsynaptic potential and the other is inhibitory postsynaptic potential. First, what is excitatory postsynaptic potential? I said so there are some neurotransmitters called excitatory neurotransmitters. These open cation channels, so there is sodium influx and there is depolarization and there is resting membrane potential goes towards depolarization, that is less negative. For if this neuron, it is from minus 65 to minus 45, that is it's moving towards less negative value. Such a potential change is called excitatory postsynaptic potential. If it is towards depolarization, it is called EPSP and the cause is influx of sodium ions. What about inhibitory postsynaptic potential? If the potential change in postsynaptic membrane is towards hyperpolarization, it is called IPSP. So, whenever inhibitory neurotransmitter is released from presynaptic neuron, it causes opening of chloride channels. Chloride is an anion. So the resting membrane potential move towards more negative value. Initially, if it was minus 65, whenever there is chloride influx, it becomes minus 70. And also there is opening of potassium channels. Potassium, as we know, it is an intracellular cation. Whenever potassium channels are opened, there is efflux of potassium. As positive charges are lost, it becomes more negative. That is, the potential is moving towards hyperpolarization, and such type of synaptic potentials are called inhibitory postsynaptic potential. A key point to remember: excitatory neurotransmitters open cation channels and inhibitory neurotransmitters open anion channels. Okay, now let us look at a different type of synapse called electrical synapse. In this type of synapse, the cells cytoplasm of adjacent cells are connected to through gap junctions, which allow the passage of ions responsible for neuronal transmission. And this type of transmission is bidirectional, while the transmission in chemical synapse is unidirectional, which is a basic property we should know in a chemical synapse. And the time for neuronal transmission in electrical synapse is very less compared to that of chemical synapse. Now let us look into the properties of synapse. Synapse have certain properties. Out of that, first property is law of forward conduction. That is, an impulse through the synapse travels only in one direction, that is unidirectional, from presynaptic to postsynaptic neuron. Second point, law of convergence, or many to one projection. Many presynaptic neurons will project to a single postsynaptic neuron. That is, presynaptic neurons are converging on single postsynaptic neuron. Next point is law of divergence. Here there is many to one projection. That is one to many projection. So one presynaptic neuron diverges and projects to multiple postsynaptic neurons. It is diverging. Third point is synaptic delay. Delay means time lapse. Here it refers to the time lapse which occurs between arrival of impulse in the presynaptic neuron and its passage to the postsynaptic terminal, where this time is lost. This time is taken for releasing of neurotransmitter into the synaptic cleft and diffusion of this neurotransmitter to postsynaptic membrane, and this action of neurotransmitter to bind to the receptor and to open ion channels. Certain time is lost, and this time lost is called as synaptic delay. It is the time lapse occurs between arrival of nerve impulse at presynaptic membrane and its passage to postsynaptic terminal. Remember the four causes. Next point is synaptic inhibitions. There are four types of inhibitions. First one is direct inhibition. Here the inhibition occurs through formation of inhibitory postsynaptic potential. That is, whenever there is inhibitory neurotransmitter release, it forms IPSP, and this type of inhibition is called direct inhibition or postsynaptic inhibition. Second type, or indirect inhibition. Here inhibition occurs when passage of impulse is inhibited by another earlier impulse. An impulse will inhibit the presynaptic neuron, so that presynaptic neuron will inhibit the postsynaptic neuron. So it is called presynaptic inhibition. I'll show you a diagram. See, here the neuron C is inhibiting neuron A. This neuron A is a presynaptic neuron to neuron B. So whenever C inhibits A, the presynaptic neuron, will inhibit the postsynaptic neuron B. This is called indirect inhibition or presynaptic inhibition. The causes for it are by opening of chloride channels or by opening of potassium channels, and by directly blocking calcium channels in the presynaptic neuron. It causes indirect inhibition. Next type is feedback inhibition. It is also called as Renshaw cell inhibition. Here, see, these two orange color neurons are alpha motor neurons in the spinal cord. See, this sends a collateral to another neuron called Renshaw cell, which is an inhibitory interneuron. This will in turn inhibit the alpha motor neuron. See, their collateral from spinal motor neuron ends on the inhibitory interneuron, which will in turn inhibit the discharge of same neuron. So it is called feedback inhibition or Renshaw cell inhibition, as the inhibiting neuron's name is called Renshaw. Okay, next is feed forward inhibition. Here, see, it is seen in cerebellum mainly. See, this Mosi fiber will directly stimulate the granular cells and Golgi cells. This granular cells, through its parallel fibers, will stimulate Purkinje cells. But the output of Purkinje cell is inhibitory in nature, and this mechanism is called feed-forward inhibition. That is, stimulation of Morse fiber will result in the inhibitory output of Purkinje cells. This mechanism is called feed-forward inhibition. Next, moving on to the next point, summation. Summation literally means to add up. Whenever there are many stimuli or impulse, the potentials are added up. That is what is called summation. First type of summation is called temporal summation. It occurs when repeated stimuli are applied at very short intervals before the EPSP due to a previous stimuli has decayed. Whenever we are giving a first stimuli, before the EPSP is getting decayed, we are giving a second stimulus so that the EPSP of first and second will add together. Similarly, when we give a third stimuli, the EPSP of all the three will be adding together. This type of summation is called temporal summation. Next type is called spatial summation. It occurs when postsynaptic membrane receives impulse from large number of presynaptic neurons simultaneously. For example, a neuron A is having 50 presynaptic neurons. When all these presynaptic neurons are fired simultaneously, the EPSP to all the presynaptic neurons will add up, and this type of summation is called spatial summation. And this graph, whenever more number of presynaptic neurons are creating an impulse, the potential reaches the threshold and even action potential may be fired. Okay. Next, moving on to next, this is occlusion. Let me explain you that first before going to the definition. See, in this picture, A is a presynaptic neuron. Whenever it is stimulated, it stimulates 5 postsynaptic neuron. And whenever B, a presynaptic neuron, is stimulated, it also stimulates 5 postsynaptic neuron. So total, when A is stimulating 5 and B is also stimulating 5. So we adding A plus B, it gives 10 neurons are stimulated when they are stimulated separately. But when A and B are stimulated simultaneously, only 8 neurons are stimulated. 8 postsynaptic neurons are fired because there are 2 neurons which have common discharge zone. It is fired even in the presence of A or in the presence of B. There is overlap of discharge zone for these 2 neurons. This phenomena is called occlusion. That is, whenever it describes the stimulation in which response to stimulation of 2 presynaptic neuron is less. That is, whenever A and B are simultaneously stimulated, the number of neurons firing is less when compared to the sum total response obtained when A and B are stimulated separately. When A and B are stimulated separately, it gives 10 neurons, but when they are simultaneously, it gives only 8 postsynaptic neurons. That is, 2 neurons have a common discharge zone, and this phenomena is called occlusion. Next phenomena is called subliminal fringe. Okay, look at the neuron A, afferent neuron. It has many branches, and it is going to act as a presynaptic neuron for many neurons. And B is similarly is an afferent neuron, and it is a presynaptic neuron for many neurons. When a neuron A is alone stimulated, it is having a discharge zone of 5 neurons. That is, it stimulates only 5 neurons of the postsynaptic neuron. When B is alone stimulated, it also will stimulate only 5 neurons. But when A and B neurons are simultaneously stimulated, it will stimulate 12 neurons. That is, 2 neurons are stimulated extra in this case when they are stimulated simultaneously. This phenomena is called subliminal fringe because the 2 extra neurons that are stimulated here because of A and B are stimulated together. Previously, when stimulated alone, A neuron is stimulated alone, it does not reach the threshold level for firing an action potential. But when both the neurons are stimulated, it receives presynaptic connection from both the neurons, and this will reach the threshold level and action potential is fired only when both A and B are stimulated. But either of them are stimulated previously, the potential is not reaching the threshold level, it only reaches the subthreshold level, so that 2 neurons are not fired. So these neurons are in the subliminal fringe region. When both A and B are simultaneously stimulated only, they have the action potential. This is called subliminal fringe effect. Next is synaptic plasticity. Plasticity means being easily modulated. That is, it can be easily modulated. There are 5 plastic changes in synaptic transmission. First is post-tetanic potentiation, long-term potentiation, synaptic fatigue, sensitization, and long-term depression. We'll discuss each of them in detail. First is post-tetanic potentiation. Here, post-tetanic potentiation, let me give you an example. We are giving a first stimulus to a neuron. Then we are giving a continuous stimulus, for example, 50 stimulus per second. So during 50 stimulus per second, there is increased calcium in the presynaptic neuron because more voltage gated sodium calcium channels are opened and calcium in the intracellular site is increased. So when we are giving next stimulus following this 50 stimulus, the postsynaptic potential is exaggerated because increased calcium will cause increased release of neurotransmitter, and that will increase the postsynaptic potential, the next stimuli. So it is due to increased calcium in the presynaptic nerve in response to arrival of stimuli that causes increased transmitter release. The postsynaptic potential, just after fraternizing potential is given, the potential is more. Next is long-term potentiation. When the post-tetanic potentiation is staying for longer time, it is called long-term potentiation. It occurs due to an increase in intracellular calcium in the postsynaptic neuron, but in post-tetanic potentiation, the calcium level is more in presynaptic neuron. It is very importantly taking place in hippocampus as it is a part of memory. Next is synaptic fatigue. Fatigue means getting tired. So there is gradually decrease in neurotransmitter release in response to repeated stimulation of impulses. The causes is inactivation of calcium channels due to repeated stimulation and exhaustion of neurotransmitters. Since there is repeated stimulation, the neurotransmitters that are stored gets exhausted. Next is sensitization. Sensitization is nothing but when you pair a stimulus with a noxious stimulus, that is a painful stimulus, there is prolonged occurrence of postsynaptic responses. Okay. Next is long-term depression. It is just opposite to the long-term potentiation. Here there is decreased synaptic conduction due to low stimulation of presynaptic neurons, and there is decreased calcium influx. This is the reason for decreased synaptic conduction. Now let us look about neurotransmitters. Neurotransmitters are nothing but they are chemical substances that are released by the presynaptic neuron into the synaptic cleft during synaptic transmission. And this release of neurotransmitter is described as quantal release, and this quantal release of neurotransmitter into the synaptic cleft is called Dale's phenomena. To see the classification of neurotransmitters. Neurotransmitters are classified based on their chemical structure. First one is acetylcholine. Second class is amines. It include norepinephrine, epinephrine, serotonin and dopamine. The third group are amino acids, under which comes glutamate and aspartate, which are excitatory amino acids, and glycine and GABA are inhibitory amino acids, inhibitory neurotransmitters. The next group is polypeptides, which includes ADH and oxytocin. Next group is purines, which includes adenosine and ATP. Last group are gas, which include nitric oxide. That's all about neurotransmitter. It is also an important question, important short note. Thank you.